for introduction. So firstly, I would like to thank the organizing committee and especially Chenkova Sensei for inviting this nice conference, for giving me a good opportunity of talking about our recent research. And today, I would like to talk about polymer structure in water studied by THz-raman spectroscopy. And this is an outline of my talk today. Firstly, I'd like to talk about brief introduction of low-frequency Raman spectroscopy for polymer research. And then I will talk about hydrogen bonding between collagen model peptide and water. And finally, I'd like to talk about 3D network polymer and water interaction. And the low-frequency Raman region probe the same low-energy vibrational mode of molecular structure as THz spectroscopy. And the low-frequency mode can give detail on the conformational changes in lattice structures, boson peak of amorphous glassy polymers, and main chain according vibrations, and higher order structure of polymer, and so on. And this is one of the good example of THz spectra of biodegradable polyester. And as you can see, this figure, the amorphous and the crystalline state of this biodegradable polymer show a quite different spectra in the low-frequency region. So the low-frequency region spectra is very sensitive to the higher order structure of the polymer structure. And we use the two kinds of Raman spectrometer for this research. By using ONDAX Raman system, you can measure the Raman spectra from 20 to 1500 wave number region. And firstly, I would like to talk about the low-frequency Raman study of the collagen model peptide. And the collagen is composed of glycine, proline, or hydroxy proline lipid. And it forms a triple helical structure in the temperature dependent. The triple helical structure is formed at low temperature, and but it dissociates at high temperature like this. We use the collagen model peptide, glycine, proline, glycine, PPG10 and PPG5 as a sample in this research. And PPG10 can form the triple helical structure like a collagen in solution in low temperature. However, the PPG5 is not able to form the collagen triple helical structure because of the length of the molecular chain of the PPG5 is too short, and this structure change of PPG10 is reversible. And at first, we compare the temperature dependent Raman spectra in the high frequency region for PPG10 and PPG5. At the high temperature, both of these collagen model peptide formed random coil like this, random coil structure, and the peak at 1655 and 1639 wave number appeared. When it cooling down, only PPG10 solution showed the three peaks at 1667, 1644, and 1628 wave numbers. according to the assignment, which is already reported, these three bands indicate triple helical structure of the collagen model peptide. As you can see from these figures, only the PPG10 form triple helical structure, and the transition temperature is at around 50 Celsius degree. And this is a low frequency region of Raman spectra of PPG10, PPG5, and D2O in the cooling process. And to make clear the structure changes, the different spectra were obtained. And here you can see the temperature-dependent spectra of different spectra of PPG10 and PPG5 and D2O. Around 170 to 180 wave number, there is a peak in the PPG10, 5, D2O, all the system. And the peak at the 170 wave number is assigned to the stretching of hydrogen bonding of water. And this peak shows shift to the higher wave number, a higher frequency region with cooling process, and because of the hydrogen bonding becomes stronger. In case of the PPG5, here, the peak at around 180 wave number shows slightly shift to a higher frequency region during cooling process because of the water does not form tetrahedral structure and hydrogen bonding between PPG5 and water becomes stronger. On the other hand, the peak at the peak of the PPG10 shows a shift to the higher wave number. Then the peak shift to the lower wave number in the cooling process. So when PPG10 form the triple helical structure, the CO group, which in the outwardly of the triple helical structure, and hydrogen bonding between water. So the state of hydrogen bonding is different with pure water. So this phenomena comes from this result. And other characteristic bands may be due to the collagen model peptide. For example, the peak at the 250 here and here wave number is appeared clearly in the PPG5 in all the temperature and PPG10 at the high temperature. So it may be due to the random coil structure of the molecules. In conclusion, for collagen model peptide, by using low frequency Raman spectroscopy, it can detect the hydrogen bonding between collagen model peptide and water, and it indicates the coil helical structure transition with temperature. And next example is the superabsorbent polymer, SAP. Yeah? So SAP absorbs 100 to 1,000 times as much water as their own weight to form gel. And SAP is composed of sodium polyacrylate, cross-linkable monomer, and polyacrylate, and it makes 3D network structures like this. And this is a Raman spectra of SAP and sodium polyacrylate. Yeah. in the low frequency region at the room temperature. And it can be seen from this figure. The band at around 126 wave number is maybe come from the SAP 3D network structures. And this is the temperature dependent Raman spectra here. And it's different spectra in low frequency region of SAP in the heating process. And as you can see here, there is a maybe two peaks in this region. And and the band at around 70 here show the shift to a lower frequency region with heating process, and the band at around 126 wave number show a shift to a higher frequency region during heating process. And this, it may be this band comes from 3D network structure of the SAP polymer, and the change of this peak intensity and peak position maybe show the structure collapse of the SAP during heating process. And we also tried to measure the additing and removing water in the SAP 3D network polymer structure. And this figure shows the Raman spectra of SAP before and after additing water, before additing water and after additing water. So totally, the first, the SAP polymer has about 5% weight water, and we added this polymer to the water, and finally we get about 20% water having polymer, SAP polymer. And this figure shows the annealing process in the vacuum oven with 100 degrees C for 0 and 10 hours and 24 hours. By adding water here, the peak position does not change so much. However, as you can see this figure, the band at around 126 wave number shows a shift to a higher frequency, like this. And by annealing some bound water in the 3D network structure of the polymer, remove from the inside of the network structure, then the part of the polymer network structure becomes shrinkage. So it may be considered the peak at 126 wave numbers show a shift to a higher frequency to the time in the vacuum oven. So finally, I'd like to show the pressure dependence of the SAP. So this is 0 megapascal here and 8 megapascal pressure to the SAP polymer with water. So unfortunately, now we have not clear in the process of the pressure dependence of the SAP, but now we can get that intensity changes of the SAP of the Raman spectra of SAP during the pressure dependence. Okay. So finally, I'd like to thank the Dr. Hoshina-san Hoshina-san and Dr. Yasuda-san in Kansei University, and also Professor Ozaki Sensei, Kansei University. And thank you for your kind attention.
Thanks, Professor Sato. So now for the discussion. Any comments or questions?
Thank you very much, Professor Sato. It was so much work. I would like to, just a comment. I can see so much place for multidimensional, multivariate analysis on that data. And all the talks that I was listening to. The whole idea of aquaphotomics is if we could all go to identify bands and see those bands at different places of electromagnetic spectrum. Do you think that this might be possible? Because I saw a different pressure,
yes,
different temperature,
yeah.
So many different perturbations.
Mm-hmm.
And you have these small changes in the spectra. I mean, THz is, when you look at this, it's a smooth curve, and you see now small peaks.
Yeah.
But if you probably do a multivariate analysis, maybe you will see clear positions of those bands. And then we can probably continue and try to see relevance between these bands and other similar information from other regions.
Yeah, I think so. So we try to.
Any other question, comment? So thanks again, Professor Sato.